Hey guys, welcome back. We're now moving on to chapter 2, which is all about cells. We're going to start off with part 1, which looks at cell organelles, and we're going to talk about the different ones. We're going to talk about their functions, all that good stuff. All right, so without further ado, let's get started. So first off, we should probably mention that there's two main classifications of cells. There are prokaryotic cells like we see here in the bottom left of the screen. And these are very, very simple. They don't have a nucleus. In fact, they have no membrane-bound organelles at all. So you notice that it's pretty simple what's going on the inside. The DNA is circular and it's just sitting right in the cytoplasm. So what it means to be circular is the ends of it actually connect and fuse together, and then it twists itself up that way in order to keep it from getting disturbed or tangled or messed with in any way. So prokaryotic organisms are bacteria. So they are all single-celled. They are simple with no membrane-bound organelles whatsoever. As opposed to eukaryotic cells, which is here on the bottom right. So they're a lot bigger. They're more complex. They have a nucleus and membrane-bound organelles. You can see there's the rough and smooth ER. We have mitochondria. There's the Golgi. So we have a lot more going on. We have a lot more organelles, all of them with their specific functions. The DNA does tend to be bigger. It is linear, it's not circular, and it is stored inside of the nucleus. Eukaryotic organisms can be single-celled or multi-cell organisms. These would include pretty much anything that is not a bacteria. So your animals, plants, fungi, and protists are all eukaryotic organisms. All right, well, again, let's start talking about the cell. Let's start with the outside of the cell. Now we're gonna focus primarily on animal cells because this is a human genetics course. So that means that we as humans are animals. So cells are surrounded by a cell membrane. Now this cell membrane, as you can see at the bottom of the cell here, these little yellow circles with the two little tails, those are phospholipids. And the cell membrane is made up of a phospholipid bilayer. So if you look again at this picture, there's one layer there and one layer there. So the heads of the phospholipids are on the outside and their tails are pointed towards each other. You have the outside of the cell above and the inside of the cell below. And then we have that bilayer separating the two. There are also proteins embedded in it. Some of them are as channels or pumps to allow things in and out of the cell. Some of them are anchor points for these carbohydrate chains. Now these can have a number of different functions. We'll talk about a few of them in the next bullet point. Let's see. You could think of the cell membrane like your skin. It surrounds your body. It keeps all of your organs safely inside so nothing can mess with them. It allows some things in and out, but it prevents a lot of bad things from getting in. It's thin, it's flexible. It allows us to interact with the world, and that's what a lot of the function of the cell membrane is. Now, on the cell membrane, we have something called the extracellular matrix. So if you look here at the other picture, we have the membrane, we have these proteins, and then we have the carbohydrate chains hanging off. And these carbohydrate chains can interact with these other fibers that are outside of the cell, which is what makes up the extracellular matrix. Now, this extracellular matrix has a few different jobs. It allows for the cells to stick together. You notice I can't just wiggle my finger and push the skin cells apart so I can see my muscles, because all the skin cells are stuck together. And it's because of this extracellular matrix that they actually grip each other and hold on, so that I can't just, and a whole section of my skin goes flying. thankfully. That'd be rather disturbing. Precise sequence of interaction between cell surfaces. So what that means is that they also interact with each other cells in terms of the cells. So they don't just hold each other, they interact. So they can pass messages along. They can recognize, if this skin cell here recognizes that it's no longer touching another cell on one side, it can actually create a chemical signal that alerts your immune system that there's a breach in the skin. Send the antibodies, send the white blood cells, send the red blood cells, send things to take care of that. And then it'll also trigger mitosis, which is cell division, in order to allow for that hole to be closed up. And cell adhesion molecules, also known as CAM, so they guide the white blood cells to the injury site. So remember how we said they can kind of send messages? As they're sending, some of the messages could be, white blood cells, keep going, stop right here. This is where the injury is. This is where you're needed. So they help to guide the white blood cells and the antibodies to where they are needed in order to prevent infection and fix an injury. They can also recognize other cells as self, as part of the organism. So our cells recognize each other. A lot of it, again, these carbohydrate chains and how they interact with the extracellular matrix. It allows them to recognize molecules, proteins, carbohydrates, lipids that our body makes and then sends throughout, or that our cells make and send throughout our body. It helps, this extracellular matrix helps to identify them and recognize them as self. So that knows not to send a message to the immune system, be like, hey, send the white blood cells. All right, let's talk about the nucleus. So the nucleus is this organelle right here. A lot of times we think about it as the control center of the cell. It holds the DNA. It holds our genetic information. So here we have the nucleus, and then you can see the chromosomes, whether the DNA is in chromosome or chromatin form, it's all stored within there. The DNA is surrounded by a nuclear membrane, and this membrane is even more selectively permeable than the cell membrane, meaning it's even more specialized in terms of what can go in and out of the nucleus. Because again, you don't want things messing with your DNA. You'll notice that there are these holes in it. These are the nuclear pores. These are the sites where things can enter or leave the nucleus. And in the center of the nucleus, we have an organelle called the nucleolus. So the nucleus doesn't just hold the DNA, it also holds the nucleolus. And this is an organelle that produces ribosomes. Once it produces ribosomes, the ribosomes will then lead through the nuclear pores in the nuclear membrane and will go out into the cytoplasm. So around the nucleus, we've mentioned our ribosomes. They are created inside the nucleus, specifically by the nucleolus, and then they can leave the nucleus. These are little itty bitty tiny tiny organelles whose job it is to make protein. So they assist and are the site of protein synthesis. They don't have a membrane, but they might be attached to the next organelle, which is the endoplasmic reticulum. Now, what the endoplasmic reticulum, or the ER, is a network of tubules and sacs made out of membranes. So if you look at this picture on the right, you can kind of see how it even connects up with the nucleus and the nuclear envelope, the nuclear membrane. Part of it is called the rough ER, and part of it is called the smooth ER. And the only difference in terms of why we call them rough and smooth is because one has ribosomes on it, giving it a bumpy appearance, and the other does not, which is why it's smooth. And we'll start with the smooth ER. That's this one here. You notice it tends to be more circular and tubular as opposed to these flatter sections. So the smooth ER, there are no ribosomes attached to it, which is why we call it smooth ER. It produces lipids and steroids that your cells need to function, and that can also be sent out of the cells in order to travel throughout the body. You tend to find more smooth ER, so more or a bigger smooth ER structure in cells that can secrete and store lipids and steroids, such as your liver. Let's see, they can also store excess enzymes. So we can put extra enzymes into the smooth ER simply to store them. And they can actually be moved directly from the rough ER through the tubular connection to the smooth ER. Now the rough ER, if you notice, it has these dots on it. Those are the ribosomes. So the rough ER has ribosomes stuck to it. And if you remember, ribosomes are the site of protein synthesis. So therefore, that tells us ribosomes have to have something to do with, the rough ER has something to do with ribosomes, because rough ER has ribosomes on it. Ribosomes make proteins. Therefore, rough ER has to do something with proteins. And essentially what it does is it takes the proteins that the ribosome made and makes them into functional molecules. Once the ribosome is done assembling the amino acid chain that will eventually become a protein, it's not functional at that point. So the ribosomes will take them to the rough ER in order to modify the proteins so that they become functional working proteins. All right, energy and transport. So again, we're focusing on animal cells, which is why, if you notice, we're not talking about the cell wall. We're not talking about the chloroplast, anything like that. First off, we have the mitochondria. Kids know it as the powerhouse of the cell, but please do not tell me that as an answer for what its job is, because that does not tell me what its job is. The job of it is to make energy. Specifically, it takes glucose molecules, it breaks the bonds in the glucose molecules, releasing energy, and takes that energy, traps it as it forms ATP. ATP is kind of like a rechargeable battery molecule. It goes out into the cell and it powers every organelle within the cell. Every reaction that occurs within the cell is powered by the ATP made from the mitochondria. Interesting little tidbit of information. Mitochondria also has its own DNA. We call it mDNA or sometimes mtDNA. But what it is, it's mitochondrial DNA. It is completely separate from the nuclear DNA, the DNA inside your nucleus. Next, we have this sequence of flat, okay. People tend to say they look like a stack of pancakes, but its function, or first of all, it's the Golgi, whether you call it Golgi body, Golgi complex, Golgi apparatus, it's the Golgi. And its function is synthesis, storing, sorting, and shipping materials. I like to think of it as the post office of the cell. So what that means is, once a protein is made by a ribosome and modified by the rough ER, it can be sent to the Golgi. The Golgi is going to tag it for whatever it needs. And a lot of the purpose of those tags are, one, for the rest of the body cells and the extracellular matrix to identify it as self so that it doesn't get attacked by the immune system. It could also add tags that are essentially pretty much shipping labels to help it figure out where it needs to go in the cell. So it'll have these tags and other cells will just, let it pass by. But when that tag is recognized by a specific cell, it'll absorb it into itself and use it for whatever function. Once it has been tagged and modified and ready to go, then it'll move that molecule to one of the ends of its layers where it'll get pinched off and form what's called a vesicle. Now, vesicle are these little sacks of membranes, and they're used for storage sometimes, but definitely for transport. So these are Golgi vesicles, and we have a picture of just a generic vesicle up here. See how it's made out of the membrane? Just like the Golgi is, just like the cell membrane is. And what happens is these membranes will move until they get to the cell membrane. They will fuse with it. They will then separate and push their contents out of the cell so it can travel through the bloodstream to wherever it needs to go. Because I messed with my camera. All right, there we go. Waste removal. We have two main organelles that help with waste removal. The first one is called lysosomes. So that is right here. These are sacks of enzymes, and they help to break down things that shouldn't have entered the cell, like bacteria, or if there's excess fats or excess food, it'll help break those down too. When the cell is ready to go through apoptosis, which is programmed cell death, it is also lysosomes that will go through and start breaking apart the different organelles and whatnot within the cell. So they do have a role in destroying the cell once it is done completely with its job. There's a particular disorder called Tay-Sachs disease, and what this disorder is now the lysosomes have usually somewhere around 30 to 32 different enzymes within them. But in Tay-Sachs disease, one of those enzymes is not made properly. And it's a particular enzyme that will break down excess fatty material in the cells that surround nerve cells. Now, since it can't break down the excess fatty tissue, they just kind of build up within the cell, causing it to swell. And these are the cells that surround nerve tissue. So eventually they swell up and they strangle the nerve cell so that signals cannot go through it anymore. So what you'll see is someone who has Tay-Sachs disease, they'll start losing muscle control, coordination. Eventually they'll also have trouble eating and swallowing. They'll have trouble breathing because the signals to move the lungs and inhale and exhale stop going through. And then eventually even the signals for the heart to beat get strangled and prevented. A lot of times, unfortunately, Tay-Sachs does have a high mortality rate, and it usually kills by the age of 5, definitely probably by the age of 10. All right, let's see, peroxisomes, which we see here. Very similar to lysosomes. They are sacks of something that breaks down other things. Like I said, these are both waste removal organelles. But the difference is what are they filled with? What do they use to break down the material? And peroxisomes do not use enzymes like the lysosome does. It uses hydrogen peroxide, H2O2. So hydrogen peroxide is in the peroxisome. Now this is good for breaking down lipids, fats, bios. We tend to see higher amounts of peroxisomes within cells found in the liver and the kidney because those have a lot of filtering and a lot of breaking down of, like I said, lipids, bio, etc. All right, structure and support. Within the cell, within the actual cytoplasm, remember the cytoplasm, we didn't mention it here because you guys should know what the cytoplasm is at this point. It's that jelly-like substance that fills the cell that the organelles are sitting in. Well, within that, we have the cytoskeleton. And what the cytoskeleton is, again, let's say this is membrane, this is all within the cell. Here's the mitochondria, and you'll see these thin, stretchy bits all over the place. The cytoskeleton is like the skeleton of your body. It gives shape to the cell. it also serves as structural components to help the organelles move around. Now, there are three main components that we're going to mention. We have microtubules, microfilaments, and intermediate filaments. So again, just different ways in which they are put together and how they are stretched throughout the cell. Notice how they are attached to the mitochondria in this picture, because what can happen is the cytoskeleton helps to support the organelles within the cytoplasm, but it can also help to move the organelles to wherever in the cell they need to go. The organelles do move, and the cytoskeleton is usually one of the ways that they're able to do that. It also serves as the foundation for cilia and flagella. So a flagella is a long tail that can stretch out of the cell. Cilia is when it's covered in these smaller versions of that. And they have to be able to move in order to allow the cell to progress through its medium. And the cytoskeleton will actually stretch from within the cytoplasm through the membrane and into the flagella and cilia in order to help them achieve that movement. Kind of like in a dog. The spine, the vertebrae bones, stretch into the tail, and it's because of the bones and the muscles that attach to the bones that a dog can wag its tail. It's the same idea. The cytoskeleton stretches into the flagella or the cilia, and it's because of that extension into those that they can move the flagella and cilia. So spherocytosis, this is a defect in the cytoskeleton of red blood cells, and symptoms cause the RBCs, red blood cells, to balloon out. So instead of being that nice kind of fluffy donut shape where it's indented in the middle, they turn into these big bulbous balloons. This means that they can very easily block different blood vessels, causing clots in different organs, and that can lead to a lot of different problems. Centrioles, you'll see this right here. These are made up of microtubules, and they only function during cell division. What they'll do is they will build the spindle fibers that attach to the chromosomes and separate the sister chromatids during division. All right, let's see. And now here we have our picture. So we're surrounded by the cell membrane. We have all this jelly-like stuff is the cytoplasm. Here's our nucleus. Has the chromatin, the DNA inside, along with the nucleolus. It's protected by the nuclear membrane or nuclear envelope, and it has the nuclear pores in it that allow things in and out. Then we have our rough ER, which it modifies proteins. Smooth ER makes lipids and fats, and mitochondria provides energy. Lysosome breaks down waste. Centrioles aids in cell division. Golgi packages and ships stuff out of the cells. Now you'll be able to see that a little bit better here. You can see the particles are being grouped in the ends of the Golgi. It pinches off, forming this vesicle, which will transport it to the membrane where it'll fuse, and it'll be pushed, the contents will be pushed out. The little red dots are the ribosomes. Let's see. Here's a peroxisome. Now, to help show the difference of lysosomes versus peroxisomes in pictures, you'll notice the lysosomes, they tend to show stuff within it to indicate the different enzymes. Peroxisomes, they really don't show that. You'll notice that there's all these different lines going through the cytoplasm. That's all of the different versions of the cytoskeleton. And we can even see the green one there pushing out here where the cilia are located. All right, let's see. I think we covered pretty much all the main things. Yeah, that looks good. All right, so that concludes part 1 of the cell organelles. I will include a link to this Prezi in our description, but the additional resources won't be till the end of the chapter. But I hope this helped to explain the information. Stay awesome, stay amazing, stay curious, and above all, stay safe and stay healthy. All right, you guys. Take care of yourselves. Bye-bye.